Hey, what's up, biology students? Welcome to the virtual lecture on photosynthesis, part 2. In this video, we're going to take a closer look at the two stages of photosynthesis, those being the light reactions and the Calvin cycle. Both of these stages occur within an organelle called the chloroplast. And if you remember from our last video, photosynthesis primarily occurs in the leaves of plants, specifically in the interior of the leaf within cells called palisade mesophyll cells. And it's these palisade mesophyll cells that are packed full of chloroplasts. If we look closer at the basic structure of a single chloroplast, we first want to notice that it has several membranes, so even more than the mitochondria. So there's an outer membrane, followed by an inner membrane, and then inside the inner membrane is a space or a compartment, analogous to the matrix. And this space here inside the inner membrane is called the stroma. And it is inside this space called the stroma where stage 2 of photosynthesis occurs. That's called the Calvin cycle. Suspended within this stroma space, you can see this large dark green interconnected structure. And this large dark green structure is actually a third membrane. It kind of looks like stacks of coins that are interconnected to one another. There's a lot of surface area. And it is here within this third membrane that stage 1 occurs, the light reactions. And the reason that this third membrane is dark green is because within this membrane are many molecules of chlorophyll. And their job is to absorb sunlight during the first stage. So the name of this third membrane is called the thylakoid membrane. And again, that is the site of the first stage of photosynthesis called the light reactions. Okay, so I'm going to say just one more time just to summarize. So in this space, it's kind of hard to point to, but it's the space inside the inner membrane that surrounds the thylakoid membrane. That's called the stroma, and that is the site of the Calvin cycle. The light reactions, which are the first stage of photosynthesis, occur within the thylakoid membrane, within these dark green structures. This slide here just summarizes both stages of photosynthesis together. And we're going to start with the light reactions. So in summary, during the light reactions, we're going to use water and sunlight to produce ATP, NADPH, and oxygen. Now, rather than just having me read a couple of paragraphs here describing the light reactions, I think it's much more beneficial for me to draw this out, and then we'll come back to this verbal description after. So we're not going to go into as great of detail in photosynthesis as we did in cellular respiration, primarily because they're very similar and it would be somewhat redundant. But photosynthesis is just as complex, if not more, than cellular respiration. Well, let's try to simplify it here. So what are we looking at? If we go back here, what I've done is I've zoomed in on that third dark green membrane. I've zoomed in on the thylakoid membrane. So you can see that here. Let's label it. All right, so this is the thylakoid membrane that was dark green in the previous picture. So the solution outside of that membrane is called the stroma, and the solution inside of it is called the thylakoid space. This is the innermost compartment of the chloroplast. Remember, I said that embedded within this thylakoid membrane are many molecules of chlorophyll, and their job is to absorb sunlight. So we're going to see that happen here in the first stage. So we're going to label a few more things here. First, we're going to label what these little green circles are. Now, each of these green circles represents a chlorophyll pigment molecule. We're just going to say chlorophyll. If you remember from the previous video, there's two major types of chlorophyll that are involved in absorbing sunlight, those being chlorophyll A and chlorophyll B. Chlorophyll A is the most common, so we can take a moment just to label some of these. Now, the first step that happens in photosynthesis is light, which is, as we learned, a form of electromagnetic radiation, finishes its 93 million mile journey from the sun until it finally strikes one of our chlorophyll molecules. So I'm going to draw that. And remember, it's specifically red and blue light that are absorbed by chlorophyll. And what happens when red and blue light, these little particles called photons of light, strike our chlorophyll molecule, is that they bash into and they strike an electron of the chlorophyll molecule. I'm just drawing these as green circles. You can look it up if you want to. It's a really complex molecule that has a magnesium atom at the center, and an electron of that atom gets excited when it hits or gets hit by the photon. So we're going to say that when the light strikes the chlorophyll pigment, it excites one of its electrons. So I'm going to draw a little black arrow to show that its electron got excited. Boom, got boosted up. And what happens next is kind of like a set of dominoes. This chlorophyll pushes, in a sense, the adjacent chlorophyll, causing the next one to get its electron also excited, pushing the next one. Now, electrons are not being transferred between the chlorophylls. They're just kind of pushing or giving each other some motion, some energy. Until finally, they push and excite the electron of a special pair of chlorophyll pigments called the reaction center. Okay, so these two chlorophylls in the center are referred to as the reaction center, and it causes their electron to also get excited. We're actually going to draw it this time. So boom, it gets excited to a higher level. And what happens is that electron actually gets taken away from this chlorophyll molecule and, in a sense, stolen by this molecule here in purple. Now, since this chlorophyll molecule just lost an electron, it really wants an electron back. And our next question is, where are we going to get a new electron from? The answer is water. This is why plants need water to do photosynthesis. So plants, they take water and they basically split it open. They, in a sense, oxidize it, releasing a pair of electrons, releasing 2 protons, and importantly for us and pretty much all heterotrophs, in the process of splitting water, they also release oxygen. That is the same oxygen that exits the leaf via a stomatal pore and dissipates into the atmosphere. Now for the plant, this step is very important because it's this pair of electrons right here that we just drew. They are going to, in a sense, replenish the chlorophyll molecule that just lost a pair of electrons, or just lost an electron. And then that cycle is going to happen again because you constantly have sunlight striking chlorophyll pigments. So the same thing's going to happen. Light's going to hit this chlorophyll molecule. They're going to push that energy to the reaction center. Boom. That electron's going to get excited and stolen. Then again, it's going to be missing an electron. It's going to take another one from water. And basically you have like a flow of electricity or a flow of electrons from water to here to here. And our question is, what is the plant going to use these electrons to do? How is it going to use these electrons to build food? Because remember, that's our ultimate goal, is to build glucose. So this is really complicated stuff, a lot of biochemistry, but I want you to understand in a simple sense why a plant needs water to do photosynthesis. And the reason it needs water is it needs to split open that water as a source of electrons. And these electrons here don't have much energy. But once they get boosted or excited using the energy derived from sunlight, they get boosted up to a higher energy level. So once that electron gets here, it now has high energy. And we're going to use that energy to do a few things, both to build glucose and to produce some ATP in the process. So the next step that this electron takes, it's going to be hard for me to circle that, so I'm going to use a different electron, pretending that we're talking about this one right here. It's then going to get transferred to a series of molecules, and it's going to get passed down like this. What does this remind you of, thinking back to the last module? Kind of looks like the electron transport chain, and it is in fact an electron transport chain. And what happens is that electron gets passed down the chain is that these molecules are going to pump protons across the membrane, as you can see here, generating a really steep concentration gradient, which is therefore used to build ATP. Now, I'm not drawing ATP synthase because I don't want to go through the ETC and chemiosmosis again. We already covered that. So we're just going to say that that electron is used to build ATP using an electron transport chain. So we're really going to simplify it. But let's take a moment to label this. And we want to emphasize what we're getting out of this process. We're basically allowing this electron I'm going to grab another electron. We're allowing this electron that has high energy to release its energy as it gets passed down the chain, allowing us to pump protons. And then remember, ATP synthase is that special protein that uses a proton concentration gradient to build ATP. So all that happens using the power of that excited electron that was excited by sunlight. Now, that electron ends up, as I drew, over here. But before I describe the last step of this process, I want to label one more thing, which is this entire structure, collectively, all of the chlorophylls. It's referred to as a light harvesting complex. We call it a photosystem. Specifically, it's called photosystem 2. Really, the overall job of photosystem 2 is just to use sunlight to energize or excite an electron to a higher energy level. And we're going to use that excited electron to produce ATP. Now that's great, we produce some ATP, but isn't the whole goal of photosynthesis to produce glucose? And so far, we haven't done that. So what's next? Well, the next destination of this electron is another photosystem called photosystem 1. We're going to take a moment to label it. And specifically, it's going to be located at the reaction center of photosystem 1. And the reason we have another photosystem is our electron, this is the same electron that started here, went here, here, and now we're here. This electron, which was excited, we used a lot of its energy in the ETC to produce ATP, so now it's back to a low energy level. So we have this photosystem 1 to boost it back up to a higher energy level. We want to excite it again. So same idea, light is going to strike this photosystem, that energy is going to get passed to the reaction center, kind of like a line of dominoes falling on each other. And then finally it's going to hit that electron and boost it to a higher energy level. And the last step is that electron, this one right here, gets taken away and collected or harvested by this molecule called NADP+, which is analogous to NAD+ from cellular respiration. It's an electron shuttle. So again, you're seeing multiple electrons here, 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 and here. I just can't erase these as I go because it will erase other stuff. So this is the same electron that we're talking about. We're just following its journey through the thylakoid membrane. So this is its final destination. And once you have a pair of electrons completing this same journey, your NADP+ is converted into NADPH. And that is our final product of the light reactions. So that was confusing. I know it was. A lot of details, a lot of structures you're unfamiliar with, a lot of chemistry. I want you to know the big picture ideas. So I want you just to know the overall products of this process and where it's occurring and why we're doing it. So let's try to summarize all of that together. First off, what are the major products? Well, there's 3. Oxygen is one of them. Now the plant can use a lot of that oxygen within its mitochondria to help it break down food, so I certainly don't consider it a waste product from the plant's perspective, and it's certainly not a waste product from the perspective of other organisms on the planet, because we really need that oxygen to breathe in to help us do cellular respiration. So that's one of the products. The second product was the production of ATP. Now, it's not just one molecule of ATP. I don't have a number off the top of my head, but we'll say several molecules of ATP. And lastly, we produced NADPH. So in summary, those are the products Our reactant was water, so that's what we came in with. Second question, broad question, where is this occurring? This is occurring inside an organelle called the chloroplast. And we said if you really zoom in to the chloroplast, if you go inside the outer membrane, inside the inner membrane, there's this collection of kind of looked like dark green stacks of coins, right? This complex third dark green inner membrane called the thylakoid membrane. That's where we're at right now. That's where this whole process is occurring, along or embedded within the thylakoid membrane. So that's where. Third, and most importantly, why is the plant doing this? How is this going to help us produce glucose? The answer to that question is this. We just produced high energy electrons that are now being carried by NADPH. In stage 2, the Calvin cycle, which we're going to talk about in a moment, NADPH is going to transfer these electrons, in a sense, to carbon dioxide, thereby building a bigger molecule, glucose. So we're going to use these high energy electrons to form new bonds and to build a bigger, more complex molecule. So in order to generate electrons with high energy, we had to do this whole process of exciting them using sunlight. Because the electrons that we started with, that originated from water in the soil, they were low energy electrons. We had to excite them or boost them to a higher energy level using the energy of blue and red light. So this is the most important product of the first stage from the plant's perspective. Okay, now if we come back, these paragraphs should make more sense. I'm not going to say this because I pretty much just said that. The reason we needed water, again, was a source of electrons. So we split open water, providing us with a source of low energy electrons, which we excited using sunlight. In the process of splitting water, oxygen is produced. So that's how we got our oxygen. That's it. I really like this cartoon picture here. That it's kind of an analogy of the more complex drawing that I just took you here, but it gets all the major points across. This is pretty much what a photosystem does. It brings an electron to a higher energy level. All right, we're ready for stage 2, which is called the Calvin cycle. Now we're going to use the ATP and the NADPH that we produce during the light reactions to help us build CO2, carbon dioxide, into glucose, food, a carbohydrate. So this is also a very complex set of biochemical reactions, but we're going to keep it simple. We're not even going to draw out a picture for it. We're just going to talk about it. But basically what happens is, as I said before, those high energy electrons that are being carried by NADPH are added to CO2 to help build a bigger molecule. ATP provides some energy to help us build. And the product is going to be basically half of glucose. It's 3 carbons. Each carbon is a purple dot. And if we put 2 of these molecules together, we have a molecule of glucose. So I'll say that again. The CO2 that is, in a sense, inhaled into the leaves via the stomatal pores, we take our ATP and our high energy electrons carried by NADPH, and we transfer them to CO2 multiple times to help us build a bigger molecule until we eventually have glucose. Then, once the plant has that glucose, it can transport it to the mitochondria and break it down to produce a large amount of ATP. It can convert that glucose into sucrose. It can store excess glucose as starch. It can also use that glucose to build cellulose, which is needed for plant cell walls, for instance. So that's it for the Calvin cycle. What is the major product of this whole process? Well, it's the major product of all of photosynthesis. It's glucose, which is food. Where does this occur? If you remember back to my initial diagram of the chloroplast, this is occurring within the stroma, which is the compartment or the space that surrounds the thylakoid membrane, but is inside the inner membrane. So this is occurring in the stroma. Why are we doing it? Well, we're doing it to produce glucose, to produce food, which is the reason that we're doing all of photosynthesis. So we're actually almost done here. A few more points about the Calvin cycle, stage 2. This first reaction here is very important. This is the reaction that brings a gas, CO2, from the atmosphere into food so that we can use that carbon for carbohydrates, for fats, for proteins, for all biomolecules. We wouldn't have carbon for all of our biomolecules if we didn't do this step. Not we, but plants. And this first step of the Calvin cycle is called carbon fixation. It's the process of taking carbon dioxide from the atmosphere and converting it into a biomolecule, which will help us build glucose. So this is how carbon enters all ecosystems on the planet. Importantly, making food available for all living things on the planet. We refer to this process of carbon fixation. It's also known as primary production. Then I have one last slide here, which is just a little bit about the Calvin cycle, a little bit more. So it says the Calvin cycle, a chaparral and desert connection. So I want to point out that some plants have adapted themselves to really dry, arid, terrestrial environments. And the way that they've done so is by only opening their stomatal pores during certain parts of the day. So think about our native plants. They're really well adapted to hot, dry conditions. So plants in the desert, plants in the coastal sage scrub, plants in the chaparral. Most of these are considered what we call cam plants. And it's these plants that are able to minimize the loss of water because they only open their stomatal pores during the nighttime. And they keep them shut during the day so that they don't lose water during the hottest part of the day. So that's kind of cool, kind of unique. They basically breathe in their CO2 at night, and then basically they store that CO2 in an organelle called the vacuole. And then it stays there in that organelle until daytime. And then when it's daytime, when there's light out, then they do both the light reactions and the Calvin cycle at the same time. So one more time, these cam plants, which includes pretty much all cactuses, succulents, and most shrubs in Southern California, they are adapted to a dry environment because of their ability to only open their stomatal pores and only take in CO2 during the night. And then during the daytime, their pores are closed. And that allows them to not lose water via those pores. Now, all other plants that are not cam plants, they traditionally just take in CO2 during the day, and they open their pores during the day. But those are usually plants that are in more temperate or tropical environments where water is not limiting. So that's it for photosynthesis. Thank you for watching.